Hey there guys, it's Justin back in your microbiology laboratory, and we're going to continue with the unknown project of separating two unknown microbial species and being able to narrow down our identification of them. We have successfully isolated them. In the last video, if you remember, we put the separated species each onto different reserve and working slants. So that's what I'm using here, are the working slants, as opposed to an old Petri plate that has been growing and aging. We want to keep using fresh cultures for all of our tests because we'll get the best, most reliable results from those. So we made reserve and working slants. I'm using the working slants here. You can see this working slant with our white looking colony and this working slant with our clear or off-white colored colony. And I went ahead and used these slants to do a gram stain, and I was able to confirm that they're both gram positive and gram negative. So the white looking one is gram positive cocci, where the other clear or off-white looking one was a gram negative rod. So under the microscope I could not only tell the gram type but also the shape of the individual cells themselves, which you guys were able to do previously this semester when you guys did your gram staining exercise. So you should know what those look like. So now I'm going to just call this gram positive and gram negative. And you can call them positive, negative, A or B, whatever you want. I'm going to name them positive and negative from here on out. And so we want to go further in this project. So now we know we've separated them and we've identified them as gram positive and gram negative, but those are very vast categories as well. So we have to keep going further down the line on identification. So next up is the utilization of carbohydrates or sugar metabolism for each of these microbes, and that'll help us further identify what uses what sugar. So what we have here are phenol red broth tubes, and I just took out some of the broth here so you could see that inner test tube in there. That's called a Durham tube. A Durham tube gets filled up with the nutrient broth as well, so we can look for bubbles inside of the Durham tube, and that signifies that there's gas production when this microbe ferments a certain sugar. So we have glucose and lactose phenol red broth tubes. So the phenol red, what that does is it's a pH indicator. So right now at a neutral or a basic pH, it's gonna be this red or pink color. And that's with nothing being fermented. So when a microbe ferments a sugar, for instance, it's going to usually leave an acid byproduct, and that's going to change the color of the phenol red indicator to yellow. So after we inoculate these today and incubate them at 24 hours, give them a chance to ferment the sugar or not, we're going to look for a color change. So if we see a yellow color change or even an orange color in these, we can say that they have fermented the specific sugar. And then we're going to look at our Durham tube too, and if we see some bubbles in there or some displacement of the media within that smaller inner tube there, that'll be positive for gas fermentation, or gas production from the fermenting sugar. So I'm going to show you how to properly inoculate these. It's pretty easy. All you need is a Bunsen burner, which I have going right here, and an inoculating loop right there. And we're just going to do one of each of glucose and lactose for each of our gram-positive and gram-negative microbes. Okay, so I'm going to start with our gram-positive here, and I'm going to do a glucose and a lactose. There we go. So when you're working with the slant tubes, we're going to remove the cap, and then I'm going to flame the lip of this on the Bunsen burner just to decontaminate anything that's on the outside of it. And also, obviously, I'm going to sterilize my inoculating loop in the flame. Okay, and I'm going to hold that in one hand like this. My screw cap is between my fingers here, and my inoculating loop is in my right hand. Okay, so now I'm going to grab one of the glucose tubes. I'm going to also sterilize the lip of this in the flame. Place the cap between my fingers over here. So my fingers are pretty full here. I have my slant here and the tube that I'm going to inoculate right here. My loop has been avoid touching anything, so it is sterile, and it's ready to go into my slant and pick up some cells. So remember, with bacteria, you don't need a lot. So I don't need to go in here and dig up a big chunk of culture there. I just need to go in here and just touch my top streak there that I have a little bit, and then put it into my sterile phenol red broth tube. I'm just going to spin it around a little bit gently because I don't want to force any bubbles into my Durham tube. Okay, so that's good to go. So after I've inoculated it, I'm going to pass it over the flame. I'm going to sterilize my loop. I'm going to take my cap and place it on there like that. So that's good to go. All right, I'll set that back there so I know I've already done it. I'm going to grab my lactose phenol red tube here, sterilize the tip of that tube. I've already sterilized my loop. I'm going to go back in, get some more cells, and then I'm going to inoculate the phenol red lactose. Okay, I'm going to sterilize my loop again. I can set that loop down for now. And then I'm going to sterilize the lip of this tube and replace the cap. So notice these caps are not airtight because we are working with aerobic microbes here, and we want some oxygen in there for them to metabolize with. Okay, so that's our gram-positive phenol red broth, glucose, and lactose tests. Now I'm going to put this gram-positive away, so I want to again flame the lip of the tube and then replace the cap. Since we've already have stuff growing on here and we just want to reserve this, I can completely tighten this cap because it's just going to go back into the refrigerator. Okay, I'm going to grab my gram-negative slant here, remove the screw cap, flame the lip of the tube to decontaminate, grab my loop. I'm going to flame that again just to be sure it's sterile. Okay. I'm going to grab my glucose broth here and flame the lip of that. Okay, now it's ready to go. Okay, I have a sterile loop. I'm going to go in, pick up some cells, and inoculate the glucose phenol red for the gram negative. Sterilize my loop. Sterilize the lip of my test tube there. Put the cap back on. Place that back there. Grab my lactose phenol red broth for my gram negative. Flame the loop or flame the lip of that tube. Gonna grab some cells from my slant and inoculate the lactose phenol red broth. Sterilize my loop because I'm done with it now. Sterilize the lip of this test tube. Replace the cap. Now I'm done with this slant, so I'm gonna flame the lip of it and then secure my screw cap tightly on this, and both of my working slants can go back into the fridge until my next tests. So these tubes will then be inoculated at 37 Celsius overnight. And again, I'm going to be looking for a color change in the phenol red indicator, and then bubble formation in the Durham tubes, which would indicate gas production. And we will check the results tomorrow.